So in this lecture, we're going to focus on protein synthesis, or the making of proteins in cells. And we're going to talk about those nucleic acids that we just finished up discussing in lecture 1. We're going to talk about how the mRNA and the tRNA and the DNA is utilized in this process. Now in protein synthesis, there's two main steps. You have transcription and the second step is translation. You need to remember that transcription happens first, translation happens second. If you don't remember which T word goes with what's happening first, then try to remember they go in alphabetical order. Transcription comes before translation. So transcription happens first in cells, and then translation happens second. Now, I'm just giving you a big idea of what's happening in transcription translation, and then we're going to focus on the details of each of these processes. In transcription, the DNA gene is being copied, just the gene, not the entire DNA molecule. The gene is being transcribed to mRNA, and this takes place in the nucleus. So we can see here that the DNA is being split open. So you see it being split open right here. We have the mRNA being made. So this is transcription, and it's taking place in the nucleus. Now, once the mRNA is made, it's going to leave the nucleus, and then translation is going to happen. So let's add that during translation, the amino acids are going to be assembled into a polypeptide, and this happens at a ribosome. We've learned that ribosomes make proteins. They make proteins by assembling amino acids into a sequence, creating this polypeptide. Now, when we get into transcription, then I have a feeling you're going to start to mix up ideas related to transcription and what we previously talked about, replication, in the last unit. Remember, we said that replication occurs during S phase because we're trying to make an entire copy of the DNA molecule. So there's two sets of DNA molecules, one for each daughter cell. So if you remember, replication is copying the whole DNA molecule. So we go basically from one ladder to two ladders. So we have a set of DNA for each daughter cells. Now, protein synthesis and transcription that happens in protein synthesis is happening all throughout a cell's life. So starting here in G1, that cell needs to make proteins, and it makes proteins by transcription and translation. So all throughout the cell's life, or throughout the cell cycle, I want to make sure you understand that protein synthesis, transcription and translation is happening all the time. It is reduced during mitosis, or actually division of the DNA in the nucleus. And that's because the DNA is coiled up into chromosomes. So the machinery that goes in and transcribes the genes on DNA can't get in there to the genes. So protein synthesis is reduced during mitosis, and that's why I stopped that arrow pretty much right here as we entered that mitotic phase. So again, that was just a general overview of the two steps of making a protein. Transcription happens in the nucleus. It results in an mRNA molecule being made. Translation happens at a ribosome, and that's where the protein is actually going to be assembled out of amino acids. Now we need to get into the specifics. So we're starting with transcription. So here I have a DNA molecule, and we can see that it is being transcribed. And just the gene, or the section of the DNA molecule that codes for this protein, is being transcribed. We are not unzipping the entire DNA molecule. That happens in replication. Now, the molecule that can unzip the DNA and make the mRNA, and you see the mRNA is being made right here, is called RNA polymerase. Let's take a second to think of the name of that molecule. So polymerase, we've got an enzyme that is building a polymer. It's building a polymer of RNA. So that's why we call this enzyme, which is being shown here by this big blob, RNA polymerase. So you can see that RNA polymerase is splitting open the DNA, and it's making a strand of RNA nucleotides by matching them up to the DNA. So if this base says T on the DNA, then RNA polymerase is going to put an RNA nucleotide that is A, adenine. If the DNA base says A, then that RNA polymerase is going to attach an RNA nucleotide that is a U, not a T. Anytime you're making mRNA and your pencil wants to write a T, you have to put a U instead, because uracil is the base that replaces thymine and those RNA nucleotides. Now, another idea that I want to point out here is you can see that there's two sides to DNA. So here's a side of DNA, and here's a side of DNA. And you can see that only one side is being copied into mRNA. The side of the DNA that is being copied is noted as the template strand. So this is the template. This is what we're copying. Sometimes it is called the coding strand. So this can be the template strand, or let's write coding strand. It is the side that is coding for the mRNA. Now, when we built DNA during DNA replication, remember the new strands are always made in the 5' to the 3' direction. And the same goes with mRNA. So an RNA polymerase is reading the DNA and building the mRNA. That mRNA is always being synthesized in that 5' to 3' direction. So if I look at this diagram here, and I can see that mRNA is being built in the middle, and I can tell this is transcription. This is not DNA replication because it's just happening to one side, just that coding side of the DNA molecule. I can tell by looking at this, since the mRNA is being made in this direction, that this would have to be, right here, would have to be a 3' end of a nucleotide. And this end down here would have been a 5' end of the nucleotide. Now we're going to get into some diagrams about transcription that are going to be new to you. Now we just indicated that there's an enzyme called RNA polymerase that can bind to the DNA at the beginning of the gene and is going to transcribe it into mRNA. So what this diagram is showing you is that from here to here, this is the coding part of DNA that is copied into the mRNA. But let's talk about this before region, the region that is before the gene or before the coding part for that protein. So let's indicate from here to here is the gene, and let's talk about this before part. Before the gene lies a section of DNA that is called the promoter region. This is where RNA polymerase binds. The RNA polymerase is going to bind to that promoter region before the gene that codes for the protein, and that's where it's going to start to separate the DNA, breaking those hydrogen bonds, and it's going to start making the mRNA. So to emphasize that, let's show a RNA polymerase molecule. So let's kind of draw a blob here and code it in. So this is RNA polymerase, and it's binding to the promoter. Now we're going to talk about the data box here in just a little bit. But first, let's relate this idea of transcription to some ideas that we talked about with cell signaling in the past. So the question is, why is that gene being transcribed? So why is RNA polymerase now binding to that promoter region and transcribing that gene? Well, let's go back to this diagram that you were shown on a previous test. And this diagram has to do with cell signaling. So remember, we have ligands or signalers, and they can bind to receptor proteins. And let's indicate this is likely a protein ligand because it cannot cross the membrane. It has to bind to a receptor protein. When it binds to that receptor protein, it activates that secondary messenger inside the cell, and it relays the signal to other molecules in the cytoplasm, like those kinases. And we can see here that during this third step, it looks like some molecule is being activated. And because this molecule got activated, then we're going to get transcription occurring. That gene is going to be expressed, and the cell is now going to make a protein because it received this signal. Now, these molecules that are often activated, that are going to promote the transcription of a gene, they're called transcription factors. So any molecule that can affect transcription is called a transcription factor. And some transcription factors are actually activators. So in this situation, since transcription is occurring after this protein got activated, this transcription factor would be an activator. But I'm going to show you that sometimes a cell receives a signal to shut off transcription of a gene. And so transcription factors are going to be activated that are called repressors. And they're going to lead to the silencing of genes, or they're going to cause transcription of certain genes to stop. Now, in the diagram above, I mentioned this TATA box. And I told you we were going to talk about that. Well, the TATA box relates to these transcription factors. RNA polymerase cannot bind to the promoter unless there are specific transcription factors that are bound to the TATA box. And you probably guess it, that the TATA box is just a section of DNA that has many repeats of AT, AT, TA, TA, and so on. So it's called the TATA box. So let's show you how the transcription factors relate to RNA polymerase in this promoter region and the TATA box before I show you some of these next diagrams that can seem very complex. So here we have the TATA box. I want to make sure you understand the TATA box is within the promoter region. So this whole area before the gene is called the promoter region. And within the promoter region, we have the TATA box. So let's show specific transcription factors being bound to the TATA box. And let's label them. These are the transcription factors. And these transcription factors can be activated by cell signaling. So I'm trying to tie together some big ideas from different units. When the transcription factors are activated and they're in place, then and only then can RNA polymerase, so let's draw an RNA polymerase, only then can RNA polymerase bind to the promoter region and can transcribe the DNA. That's what this arrow is attempting to show you, is that it's splitting open the DNA and it's making mRNA. Lots of times you'll see an arrow coming down from a gene if transcription is taking place, and they'll show you a new strand and they'll have it labeled. That's the mRNA. And the mRNA was made by RNA polymerase as it unzipped the DNA and transcribed this gene. Now let's relate those basic ideas that we just learned to this diagram and try to explain why or how cells are able to turn on specific genes. So here we have the coding region on a strand of DNA, and it's going to code for the mRNA that can later be translated. We have the promoter region that's located before the gene. We can see that we have these transcription factors in place, and RNA polymerase can now bind to the promoter region and can transcribe this gene. Let's talk about some of these other things that you're seeing here. Now, again, this is just a strand of DNA. So it's a long molecule, it's a long polymer. And different areas of the DNA that don't code for polypeptides have different functions as well. So we can see here that there's some segments of DNA that are called these enhancers. And I like to call them enhancer regions. And again, they're just sections of DNA. They're a section of bases, and that area on the DNA is called the enhancer region. We also have some areas that are called silencer regions. And I'm going to draw that on this molecule. So we have some enhancer regions, but let's say also we have some sections or segments of DNA that are called the silencers. And they're going to assist in turning off transcription of a gene. Now, we also have some molecules here that are called activators. So I just want you to think of these as being proteins that whenever the cell receives a signal, these proteins can become active. And they're kind of like transcription factors, is they're going to assist in the turning on or the regulation of this gene. Activators are what we call these proteins that bind to the enhancer regions. So these Activators bind to these enhancer regions, and they also bind with other proteins and the transcription factors that are binding to the box. When you get the right combination of activators bound to these enhancer regions and bound to these transcription factors, then the gene is going to be expressed. The gene is going to be turned on. And again, what we're focusing on is how are genes regulated? How are certain genes turned on? We have to have activators activated. They have to be bound to enhancer regions. We have to have transcription factors in place on the box before RNA polymerase can bind to and transcribe or turn on the gene. So let's show you some other diagrams related to regulating the genes of DNA, or expressing them or silencing them. This is a very similar diagram. It's just showing you DNA stretched out. So we have upstream, and I want to make sure I emphasize that word. Here we have the gene, but before the gene, or upstream on that DNA, is where we find these enhancer regions. And we can see that we have some activators that were activated, and they're starting to bind to those enhancer regions of DNA. And you can see that we have the enhancers here. So we can see the enhancers here. And they're binding to these other proteins, these transcription factors. And they're going to bind here. And again, that area was called the box of the promoter. And when those transcription factors and activators are in place, then RNA polymerase can bind to the promoter region and can transcribe, making mRNA. So in this box, I've summarized what you should be able to explain to me about how transcription is initiated in cells. Now let's talk a little bit about the silencer regions, because I haven't talked much about them, but we did draw one in. Cells can also receive signals to shut off genes. Again, we're talking about gene regulation, how genes are turned on and turned off. So when a cell receives a signal to turn off a gene, then it activates proteins in the cytoplasm that are called repressors. And repressors can bind to these silencer regions. So just like activators bind to enhancer regions, these repressor proteins are binding to these silencer regions. And whenever they do that, so let's show this repressor being bound here. And let's put REP there for repressor. Then these activators cannot bind to the enhancer regions, and then our transcription factors aren't coming together forming this complex. RNA polymerase cannot bind to the promoter region, and so transcription is stopped, or the gene is silenced or turned off. So now we're into post-transcription. So we just showed you how transcription is initiated and how this mRNA is made. So now that we have an mRNA made, then what are we going to do with it? Now, before I get into what we call mRNA processing, let's talk about this gene. So here we have DNA, and we know that sections code for polypeptides, and those sections are called genes. And we know that we make a copy of the gene, and that copy is called mRNA. Well, when we picture genes, we think that this whole area here, this whole area, this gene, it codes for this polypeptide. But that's not exactly the case. In that gene, only some sections of the DNA actually code for some of the amino acids to go in the protein. And that's why this diagram is color-coded for you. So it's showing you that here's the DNA gene that's going to code for a protein. And we can see some blue parts and we have some green parts. The blue parts are the parts that actually code for a protein. And the green parts do not. So they are non-coding. Now, these segments that do not code for amino acids, these non-coding parts of the gene, they are called the introns. The parts that do code for the amino acids of the protein are called the exons. And when we make the mRNA off of this gene, we copy both the introns and the exons, even though the introns do not code for amino acids in the protein. So what happens is the parts that do not code for the protein is going to be cut out. So another way to say that is these introns, these non-coding regions, they have to be spliced out. And that's why this is color-coded, because you can see that we're going to cut out all of these green regions, they're non-coding regions, and we're going to splice together just these parts that code for the polypeptide. So that's why this mRNA molecule here is all this pink color, and you don't see any green anymore. And then finally, this mRNA, after it's been spliced, is what's going to leave the nucleus. And this mRNA that we worked on here a little bit after transcription was over, we say that this mRNA is the processed mRNA. So this is processed mRNA. Sometimes we say that this is the mature mRNA. This mRNA sometimes is termed the pre-RNA. So if you see pre-RNA, then that means that it hasn't been processed. The introns have not been spliced out. Now, it's pretty tricky to remember which parts are going to be cut out. Is it going to be the exons or is it going to be the introns? The easiest way to remember that is the exons will exit the nucleus. The introns are cut out. They're going to stay in the nucleus. It's the exons that are going to be spliced together, and they're going to be the ones that are leaving the nucleus, like you see here in the diagram. If you notice, there's a couple other modifications that have happened to the RNA molecule. So we can see that we've added this part to it from that pre-RNA molecule, and that's going to be the cap. And the cap is going on the 5' end, so let's indicate that. And then we can see the A's on the end, and that is called the polyadenine tail. And you can just write poly A tail. And that is going to go on the 3' end of an RNA molecule. And these additions are just another part of processing the mRNA. So basically 3 things have to happen to mRNA to process it. We have to splice out the introns, we have to put a guanine cap on the 5' end, and we have to add a polyadenine tail to the 3' end of the mRNA molecule. Let's indicate that. A polyadenine tail is added to the 3' end of the mRNA. And the reason why this has to happen is because the polyadenine tail helps the mRNA leave the nucleus. It helps it get through the pores or the openings in that nuclear envelope. To help you remember what the tail aids in, which is the exiting of the nucleus, then I like to think of if you're going to go diving off of a cliff and you're not familiar with that area and you don't know how deep it is or if there's rocks in the nearby vicinity, you're taking a chance here. You do not want to go head first, right? You're going to go tail first. So it's the tail that is going to lead the mRNA out of the nucleus. So the tail goes through the opening first, and then eventually the head is going to follow out. Now that tail again is just made of nucleotides, RNA nucleotides that have the base adenine on them, and it's repeated over and over and over again. Sometimes you get about 200 of these RNA nucleotides with adenine added to the end as part of the tail. Now the cap sometimes is referred to as a guanine cap. And again, it's added to the 5' end of the mRNA. And we're going to see that that cap is important for translation. It allows that ribosome to attach the mRNA so translation can begin. So again, when mRNA is being processed after transcription is over, there's 3 things that happen to it. We have to splice out the introns, we have to add the polyadenine tail, and we have to add the guanine cap to that 5' end of the mRNA. So I just wanted to show you some diagrams that is showing you the processing of mRNA. So if you see a diagram like this, they're actually showing you the process of how those introns are being spliced out. And there are proteins that are involved, and they form a complex, which is called a spliceosome. And you can see that they're going to cut out this intron here and here, and they're going to kind of glue those 2 exon ends together. So they're splicing out the introns. Here's a different diagram over here. So whenever you see a gene and you can see that there's different parts of it color-coded or highlighted, then it's probably trying to show you that these are different from these. These are the exons. These are those non-coding introns, which do not code for amino acids in the protein. And you can see that they are being spliced out. And our exons are being spliced together. And again, this is referred to as mature mRNA. And again, here's the pre-mRNA or the primary mRNA. And you can see that we added the cap and the tail. So when you see diagrams like this, you have to note that transcription has happened and the mRNA is being processed. Now, something that I find highly fascinating is that one gene in the DNA can actually code for different polypeptides. And it can do that because of this RNA splicing that we just talked about. So here we have a gene coding for mRNA. So the mRNA contains both those introns and the exons, and we're going to splice out the introns, splice together those exons. So whenever the exons are spliced together, they can be put in this combination. And this combination is going to code for a sequence of amino acids that ends up with a protein that looks like that. But that exact same gene coding for that exact same mRNA can be spliced different. And we can see that we have a different combination of exons being spliced together there, which will code for a different protein product. So it's really interesting that the same gene in two different cells can lead to different protein. And it's because of alternative RNA splicing. And I want to indicate that phrase, alternative RNA splicing can lead to different proteins from the same gene. Now, this diagram is showing you the exact same idea that we just talked about, but again, I just want to give you different visuals. You can see that we have DNA with the exons. We make that primary RNA that's not processed yet, but when it is processed, we splice out those introns, putting together these exons here. But if they're put together in different arrangements, like this one versus this one versus this one, then whenever translation occurs, you get different proteins with different jobs, all coming from that exact same gene on the DNA. Now we're going to get into translation. So in the bigger scale of things, we are right here. So the mRNA was made during transcription. It was processed, and now it's able to leave the nucleus. Its destination is a ribosome. You can see a little ribosome right there shaded in. And this is a bigger version of our ribosome. Now there's 3 steps to translation. There's initiation, elongation, and termination. We're going to go through each of these steps. So in initiation, that mRNA arrives at a ribosome. And we see that we've got 2 parts to this ribosome. We have what's called a small subunit, and we have a bigger subunit. And the small subunit is binding to the mRNA. And the smaller subunit is going to find what's called the start codon. And you see that right here. Remember, every 3 bases, so I'm going to box in 3 bases, on the mRNA is called a codon. And the start codon is always AUG. And they're showing you that right here. So the first codon that's going to code for the first amino acid is always AUG. Now we can see that a larger subunit is binding, that's completing this ribosome. And we also can see that we have a molecule attached to that start codon, which is the tRNA. And this diagram is getting a little bit ahead of itself here, but basically the job of the ribosome is to help this tRNA match up to the codons on the mRNA. So let's take a look at this diagram here, which is trying to blow up this area of the diagram. So the codon on the mRNA, so this is the mRNA, is AUG. And the tRNA has 3 bases on it as well that are complementary to the AUG. A goes with, we have to put U because there's no T because we're dealing with tRNA. U goes with A, G goes with C. So sometimes you're asked to identify what would be the anticodon that would hook up to this codon. So again, on the mRNA, the 3 bases are referred to as a codon. On the tRNA, the 3 bases that are complementary to the codon are called the anticodon. Anticodons are on the tRNA, the codons are on the mRNA. Now let's take a look at the rest of this tRNA. This is another important part to note. This is the amino acid. Every tRNA is carrying one amino acid. This first tRNA that's complementary to the codon AUG is going to land onto the ribosome, and we have different terms for different regions of this ribosome. This part is called the E site, where the first tRNA is landing is called the P site, and then this third region is called the A site. So the first tRNA is landing in this P site here. Now all that is initiation of translation. Now we're going to talk about how we're going to elongate this strand of amino acids. We're going to build this polypeptide. And that's what the next set of diagrams is attempting to show you. So let's review what we just talked about with initiation in this part of the diagram, because this part is showing you initiation. So here is our small subunit. Let's label that. This is the small ribosome subunit. We have an mRNA molecule, and this would be the start codon. Let's indicate start codon, AUG. So we have our tRNA, and it has 3 bases that are complementary. And those 3 bases are called the anticodon. And we see that it's carrying an amino acid. And AUG always codes for the amino acid methionine. And we know that this tRNA has landed in the P site on that ribosome. So all that's initiation. Now we're ready to extend this chain of amino acids, or elongate the chain of amino acids. Now, to help you remember these sites on the ribosomes, it's EPA. That also stands for Environmental Protection Agency. But basically, you can remember that the E stands for exciting, and I like to think of the A as meaning the accepting site or the attachment site where these new tRNAs are going to land. So here we have that first amino acid in the P site, and we can see that we have the A site where the second tRNA is landing. So it's being accepted. And whenever these 2 tRNAs get together and their amino acids get close together, then it forms a peptide bond between those amino acids. From there, this ribosome is going to shift down, and this first tRNA is no longer in the P site. You can see it right here. It is now in the E site, and that E stands for exit. So you can see that it's left behind its amino acid. There's its amino acid right there, and it is now bound to the second amino acid in the polypeptide chain. So now the second tRNA is in the P site, which means the A site is open to accept another tRNA. So this tRNA is going to land, and it can match up with its complementary bases. If you notice, CGA is complementary to GCU, and this is going to be transferred here. So let's show that now we have these 2 amino acids hooked onto that third amino acid. So we are elongating the strand of amino acids. And again, we have this tRNA is going to be in the E site when the ribosome shifts down, and the A site is going to be open. So more and more tRNAs are going to be bringing these amino acids to add them to the polypeptide chain. Now let's talk about the termination. So if you remember on the codon chart, which I'm going to show you here in a little bit, there was some stop codons. So whenever you saw a stop codon like UAG, you know to stop adding amino acids to the polypeptide. So this is showing you that there are some bases or codons that are called these stop codons, and they do not code for a tRNA carrying amino acid. Instead, they code for what's called this release factor. So this release factor lands in that A site, and what it does is it's going to cut. So let's show that. It's going to cut off this polypeptide chain from that last tRNA. So now we have a free polypeptide that can fold up into a protein structure. And then we can see that the subunits disassociate from the mRNA, and basically translation is over. So that's the termination of translation. Now, one thing that I want to point out here, why we have this diagram to do this, is that as long as this mRNA is in the cytoplasm, another ribosome can bind to it and can translate it, making this polypeptide. So there are enzyme. So let's show some enzymes that will cleave up this mRNA molecule into small fragments so that it can no longer be translated, because there's sometimes a cell doesn't need to make this protein. So it has to destroy the mRNA, or the cell is just going to continuously make this protein even whenever the protein is not needed. On this page, I wanted to add a diagram of protein synthesis showing you transcription and translation just in a different way. So you should be able to look at this and have an idea of what's going on. It's just a different way to show some of the events that are taking place in the steps of protein synthesis. So again, we have transcription being taken place in the nucleus. We can see that the mRNA is being made. We know that this is mRNA. It's labeled, but you can also see that it's identifying codons. So if you see codons, you know you're talking about an mRNA molecule here. So the mRNA gets processed and it's going to leave the nucleus, and we see that a ribosome back in the distance, there's a ribosome there that is going to bind to the start codon, and it's going to continue to move down the mRNA molecule, and we can see that the protein or that polypeptide chain is growing, or elongation is occurring, and that eventually that ribosome is going to hit a stop sequence, and no more amino acids are going to be added, and that polypeptide chain is going to be complete. Now down here is that mRNA codon chart that I mentioned before, and I want you to circle codon a dozen times to try to emphasize that this is a codon chart, because here you're going to give me tRNA anticodons, and if you look up the anticodons, then every amino acid that you write here on this line is going to be incorrect. We could make a tRNA anticodon chart, but we don't. That is an mRNA codon chart. So make sure that whenever you're using this chart, you are going off of this mRNA sequence. Now we've done this lots of times in pre-AP bio, but let's do a quick practice so that you make sure that you can take the DNA and turn it into the mRNA, take the mRNA, translate it into that tRNA anticodon sequence, and then figure out the amino acid sequence. So here we have, it says the non-template strand of DNA, and down here it wants you to decipher what the template strand is going to look like. So we're just making the other side of DNA. DNA is double-stranded. So we should be using A's and T's and C's and G's. So G goes with C, T goes with A goes with T, and so on. So fill in this. And you can indicate the end since DNA is anti-parallel. If this is a 5' end, then this will be a 3' end on that A nucleotide. If this A has a 3' end on it, then this T will have a 5' end, ending that strand of nucleotides. Now, let's turn this into mRNA, and you have to note that they're saying this side is the template strand. So this is a strand that you have to go off of whenever you're making the mRNA. And again, mRNA is made in the 5' to the 3' direction. So that means that the RNA polymerase would start on this end and it would go this way. So if the DNA has a C on it, the RNA polymerase is going to attach a G RNA nucleotide. If the DNA has an A, then an RNA nucleotide with U is going to be attached next in the mRNA molecule. So T goes with A, G goes with C, T goes with A goes with U. Go ahead and add the rest. Remember, the ribosome is going to slide down the mRNA until it finds that start codon, AUG. So let's find that start codon, and it is right here. And I like to box off the codons so my frame of reading doesn't get off. So AUG is where the first tRNA is going to attach. Now, I like to draw my tRNAs when we're going through notes to emphasize that the tRNAs have 3 bases, or they have the anticodon that's going to hook up to the codon. So the tRNA would have the anticodon UAC. And I'm going to draw this tRNA. I'm going to draw it carrying an amino acid. Now, we're going to look up the amino acids here in just a second. Let's go ahead and figure out what the tRNA anticodons would be. The next anticodon would be GGU. And then the next tRNA would have an anticodon that is AUC. Now, when you're asked to look up or determine the amino acid sequence, again, you have to remember that you're looking at the mRNA codons, not these tRNA anticodons. Because again, these are the anticodons. AUG codes for MET. CCA is the next codon, and that codes for proline, PRO. And then UAG is a stop. And note, this is just a very simple model. We have only 2 amino acids in this protein, so this isn't a realistic polypeptide that we're forming here. Now let's review some concepts related to protein synthesis that we learned earlier in the school year. So we talked about attached versus unattached ribosomes. Remember, attached ribosomes are found on the rough ER. Those unattached ribosomes are just floating around the cytoplasm. You need to remember that it's these attached ribosomes, we see attached ribosomes there, that are making the proteins for 3 different purposes. These proteins can be secreted, they can be embedded in the plasma membrane, and some of these proteins end up being enzymes inside of lysosomes. So you need to remember that the secretory pathway, or the pathway for creating these enzymes that are going to be secreted, or these proteins are going to be secreted, is it starts with the attached ribosomes. They will send the polypeptide to the rough ER. The rough ER modifies that polypeptide, can send it to the Golgi, and the Golgi is what tags it and sends it to the membrane, either to insert those proteins or to release them through exocytosis. Here we see some unattached ribosomes, so they're not bound to that rough ER, and we can see that they're reading the mRNA, making a polypeptide sequence that is being released in the cytoplasm. And this polypeptide, or this protein, can be exported into different cell parts, which you see here, and sometimes they just remain in the cytosol. To give you an example of a protein that would be made in this way would be a kinase. Remember, kinases are in the cytoplasm, and they're waiting to be activated by phosphorylation by those secondary messengers. In addition, I want to take a second to revisit this idea of folding of proteins. Remember, there's 4 levels of protein: that primary, secondary, tertiary, and quaternary level. Remember that we have just that chain of amino acids with those peptide bonds forming between the amine groups and the carboxyl groups. So when we're putting together our amino acids here, and our tRNAs are bringing them close together, we have an amine group on one side of the amino acid, carboxyl group on the other. And amine groups from one amino acid are binding to the carboxyl groups of another, and that forms that peptide bond. So that's the bonding that is involved in this primary protein structure. In the secondary protein structure, we have the formation of 2 different structures, those beta pleats and those alpha helices. So here's our polypeptide chain that was assembled in translation. And so here is a carboxyl group, and here's an amine group. Here is a carboxyl group and an amine group. And the amine group and carboxyl groups of non-adjacent amino acids can form those hydrogen bonds, making those beta pleats and those alpha helices. And then we have our tertiary structure, and the tertiary structure is due to the interactions of those side chains. Remember, those amino acids have side chains that have different properties. Polar side chains can interact with other polar side chains, making those hydrogen bonds. We talked about side chains of cysteine amino acids that contain those sulfur atoms, can form those disulfide covalent bonds, which are very strong bonds. Remember that some side chains will fold outward if they're hydrophilic. The hydrophobic side chains will form inward. And then hydrophobic or nonpolar side chains can interact with the other nonpolar side chains, forming those van der Waals forces. And then finally, when we have several polypeptide chains coming together, then it makes that final structure of the protein. So again, these are ideas related to protein synthesis that we talked about earlier in the year, and I just wanted to emphasize this information.